Anhydride chemistry is beautiful and it's extremely important, but the part that's so extremely important probably isn't taught so much in introductory organic chemistry. So let me use this video to go over what you will be taught and then the hugely important part that follows from that. Let's start simply. If I take two molecules of a carboxylic acid and somehow extract water from that, I get a symmetrical carboxylic acid anhydride. It's symmetrical because there's a plane of symmetry. The two substituents on the anhydride are the same. If somehow it's possible to take two different carboxylic acid molecules and selectively remove water, then we would form an anhydride with two different substituents. It doesn't have a plane of symmetry. It's an unsymmetrical anhydride. Symmetrical ones have a plane of symmetry, unsymmetrical ones don't. Let me play a different game here. Let me take our carboxylic acid and react it with thionyl chloride. You may say that's cheating, that's not another acid, but bear with me for a second. When I do that, I get this species, which really is a mixture of a carbon acid, a carboxylic acid, and a sulfur acid. I could conceptually suggest to you that we might form this from that sulfur acid there, but actually that one doesn't exist. It goes to HCl and SO2. But if we could, it would be a mixed anhydride. It's worth remembering the structure of that mixed anhydride because it's the key intermediate in transformation of carboxylic acids into acyl chlorides by thionyl chloride. Attack of Cl- occurs on the carbonyl and not on the sulfur. HCl and SO2 are liberated. Selectivity for attack on the carbonyl derived from the carboxylic acid is useful in organic synthesis. Let's deliberately form a mixed anhydride from a carboxylic acid and ethyl chloroformate. So what we have here is a mixed anhydride from, as I say, a carboxylic acid and a carbonic acid derivative. Nucleophiles preferentially add to the carbonyl of the carboxylic acid side. And when they do, the reaction is driven entropically by formation of CO2 and, in this case, ethoxide. So if my nucleophile is an amine, I will form an amide. Now I think you can see, without me drawing it, if I form a symmetrical anhydride of a very precious acid, where I need as much of that R group as I possibly can get, then if I attack with an amine, half will go to the amide, which I might desire, but the other half will go to a carboxylate, which may not be directly useful to me. If I took an unsymmetrical anhydride, and I'll draw this one out here, attack on one side will give me an amine that I want, but attack on the other side would give me a carboxylic acid and waste amide. That's not very efficient. Mixed anhydrides, then, are very good for atom efficiency in the synthesis. And that's one of the reasons why nature uses them. It uses anhydrides like this one to transfer acyl groups. And when that happens, that phosphate is helping the whole molecule to be more water soluble. Nature uses this anhydride, this is phosphoenol pyruvate, at the start of the shikimic acid pathway. The shikimic acid pathway is fundamental to life in plants. Now let's imagine we take two phosphoric acid units and dehydrate them. We'll get a species which actually inaccurately we call a diphosphate, but it's not, it's an anhydride. If I were to take 3 phosphoric acid units and dehydrate them, I'd get a species which again, inaccurately, we call a triphosphate. But it's not. It's an anhydride of 3 phosphoric acids. Now you may say, that's not organic. I don't care. But what if we take 2 molecules of phosphoric acid and 1 phosphomonoester, where that R group on the monoester is organic? Then dehydration, loss of 2 molecules of water, gives us something we call a triphosphate, which is in fact a linear anhydride of that organic fragment. It's misleading to show triphosphates in the hydroxyl form throughout, because those OH groups will be ionized under physiological conditions. And so we'll have salts dragging the organic parts into solution in cells. I think you can see where I'm going with this, because if that organic fragment is a nuclear base, we have a nuclear base triphosphate, an NTP. And of course, the star of NTPs is ATP, adenosine triphosphate. They all have critical roles, but ATP is the one that frequently transfers the terminal phosphorus group, that's the gamma phosphorus group, to other substrates. And that is the part of anhydride chemistry which is just off-scale important. Many biochemical pathways rely on phosphorus transfer from ATP to another substrate and liberate ADP. So let me give you some examples of phosphorus transfer involving these types of anhydrides. They're all huge. So the first one is in glycolysis. The first step in taking glucose to energy is a phosphorylation. And that phosphorylated glucose goes through many steps then to generate energy. Another type of phosphorus transfer, this time giving a diphosphate residue and leaving mononucleotide phosphates, is in replication of DNA. When DNA polymerases replicate DNA as the complement, they draw on triphosphates of all the deoxynucleotide triphosphates, A, T, G, and C, liberate diphosphate units, and incorporate the nucleobases as phosphodiesters in the complementary strands. Another off-scale important application of these types of anhydrides is in cell signaling. In cell signaling, a phosphorus is transferred from a nucleotide triphosphate to tyrosine, serine, or threonine. The creation of phosphoamino acid derivatives in a protein changes its shape. It changes what that protein will interact with. Protein-protein interactions are modulated by kinase enzymes. Kinases mediate the formation of tyrosine, threonine, or serine phosphates. And therefore, they trigger sequences of protein-protein interactions, usually involving many steps of phosphorylation. And ultimately, those processes lead to an interaction near the nucleus. And in the nucleus, the cell is induced to either stop production of some proteins or start production of others. In other words, signals to cell surface receptors trigger phosphorylation reactions mediated by kinases to give signals telling the cells to grow or die in some cases. Hugely important to all branches of the life sciences, including organic chemistry. The study of kinases in medicinal chemistry has been huge in the last 10 years. The study of phosphatases, which take off the phosphorus and therefore counterbalance them, is really exciting at the moment. So I hope you can see that in organic chemistry for synthesis, symmetrical, unsymmetrical, and mixed anhydrides play a role. But in life, anhydrides of phosphorus based on triphosphate, diphosphate, and monophosphate units are pivotal. And those applications include, as I said, shikimic acid pathway in plants, glycolysis for energy production, replication of DNA by DNA polymerases, production of RNA by other polymerases, and cell signaling involving transfer of phosphorus to proteins, causing them to interact in different ways, ultimately giving signals to the nucleus directing the cell to divide, grow, or die. I hope this helps with your studies, but even more, I hope it helps you appreciate life sciences. Thank you very much for listening. Bye-bye.